So I'm Dr. Byrne. I'm Dave Byrne. I don't call myself doctor, so Dave is fine. And what I want to do is give you a little history that he already covered a little bit, but I'll go through it again about how I got into roses, because as he mentioned, I'm actually hired here as a peach breeder. And somehow I got distracted, but that's okay. It's a good distraction. And so I'm going to talk a little about the history, some of the ongoing research, just give you an idea of what we've been doing, and then we'll talk more about the Rose Rosette project and what's going on there. And this is how I got involved in roses. I came here in '83. In the early '90s, Dr. Basye came to my door and he asked me if I could count chromosomes of all things. And I said, well, yeah, of course I can. And so I did, and after that, every Friday afternoon, I go to his house, we walk through his roses, have ice cream and coffee. That was the routine every Friday. So I, spend my, usually about 2 till whenever, just talking about roses. And so he got me involved, and he was basically a backyard, a hobbyist breeder. And all the things he's released have been when nursery people went there and said, hey, that looks nice. And he said, okay, here, have it. And then it'd be released. We have Basye's blueberries, Basye purple, berry shelters. And then later, Ralph Moore came into the picture, a little more than 5 years ago. It's been a while. And I had known him because when I started working with roses, the first thing I did, since it was a new crop, I went to visit breeders to figure out, how am I going to do this? I had no idea what a rose was beyond it was pretty. And so I went to visit various breeders, and turned out a lot of the breeders there, actually the 2 major breeders in the U.S. were graduates from this department. I had no idea. And so I introduced myself, and they took me around, and then I saw Ralph as well, because he was in the area, and he was just amazing. But Dr. Basye, he was trying to solve the black spot problem, which unfortunately we haven't quite solved. But we're getting there. We're working on it. And he was all about carefree roses. He wanted something you could put out there and not worry about it, just keep producing flowers and be pretty. So that's what he was all about. And these are some of the things he released. Again, as he released them, it was basically a nurseryman that went there, took the budwood and said, okay, and he just would give the budwood away. He wasn't really in there to earn money. It was just because he loved doing it. And he did some very interesting breeding things. And so we talked a lot about that. In 2007, I got a call from actually his lawyer, Ralph Moore's lawyer, and he said, would you be interested in receiving a gift from Ralph Moore? I said, well, yeah, is this for real? You don't get many calls like that. And so he was interested in gifting all his intellectual property, all his varieties, all his selections, and the whole range of things that he developed over his 70 years of developing roses. And of course, he was really very well known. This is his 100th birthday. He died 3 years after this. But he developed roses for about 70, 80 years of his life. And when he was, until his last day, he lived on the nursery. He had a house there. He would go out and look at things and work with it. So it was just a wonderful thing, because once he gave all this material to me, I had to go out there on a regular basis. And so I could spend hours and hours with him and just talking and trying to understand things. And he released a lot of things. Over his lifetime, he released, about 600 varieties. And during much of his breeding career, he was releasing 10 varieties a year. So it's just an enormous output. And he was known as the father of the miniature rose because he did a lot of work with miniatures, but only about half his work was actually miniatures. So about 300 varieties, and you can see the wide range of miniature roses there. But he also worked on striped roses. He was one of the first people that really used it, and some of his striped roses were used in a lot of other breeding. He worked with halo roses, and these are interesting because you're talking about the dark red at the base of the petal. And that comes from a couple of different sources. But he did a lot of work, and it's been continued with other breeders as well. Moss roses, and moss rose, if you look at the bud, it has very glandular hair, so it's really very, I wouldn't call it stinky, fragrant, that's the word I was looking for, yeah. But it's more like a pine scent. It's not, you know, a traditional rose scent. It's very different. It's kind of sticky too, actually. Or crested roses, you can see the real fancy sepals it has. And it's, you know, his thought was, well, we could get this in a cut rose, and so you'd have fancy sepals and a nice flower as well. So he started that, and a lot of this stuff he started when he was about 80. You know, so he was always optimistic. And he always said, well, why not? Why can't I do that? And so he tried to do it. And so he did a lot of interesting things. He worked with Brachydes hybrids, rugosa hybrids, which are related species, trying to incorporate a lot of different germplasm into the rose background. And of course, both of these guys, this is sort of what they both said. We now need to unlock the treasures of the entire rose genus. One man, one lifetime are not enough. The logical answer is a university where the torch can be passed. Now, I used to be the younger generation when they were alive. Now I'm the older generation, so I guess I'll pass the torch sooner or later if they can get me retired. Anyways, because I'm having a lot of fun, you know, that's a problem. Anyway, so their vision was actually to set up a center of excellence for rose and ornamental breeding. So we've been slowly getting this established. Of course, graduate students are key on this, visiting scientists, and a lot of other things that we do. But when you look at the breeding program that we do, we're basically looking for well-adapted roses. And that includes disease-resistant, of course, here in Texas, heat tolerance. Because I like roses in the summertime, too. I had visitors from Japan, and they were excited about the roses, and actually they released some in Japan. And you know, I said, well, you know, they wanted me to go in May. I'm just too busy in May. I can't go jump off a plane and go to Japan for a week or two. And I said, well, how about August? And they just looked at me and said, August? There's no flowers in August. I said, well, yeah, but you just got to have the right varieties. And so we're working on that. Of course, a lot of flowers and unique horticultural traits. So there's a lot of different things there. And the reason we work on adaptation and disease resistance is this. We did a survey a while back, and we had 1,500 different responses and looked at the importance of various rose traits. And the most important one is disease resistance. It's quite a bit more important than anything else. And that's just industry people, gardeners, master gardeners, a lot of different people answered this. It's just amazing, and to see that trend so marked. And after that, of course, fragrance, flower color, density, plant size, a lot of other traits are important too. But disease resistance really stuck out. And so that's where our emphasis has always been. Of course, we have lots of good diseases. I call them good, you may not, but you know, black spot, of course, is one that's most common around here. But we're getting a lot of Cercospora, which is similar to black spot. But the symptoms are really quite distinct, but they both cause defoliation. So, you know, it just makes your plant into a stick plant, which is no fun. And of course, rust, I don't see much rust around here. It's got the orange pustules on the back. Powdery mildew in the spring, maybe a little, but it usually gets too hot for a lot of powdery mildew around here. Downy mildew can be a problem at times. And of course, rose rosette. And that's sort of a new one on the block that's getting a lot of attention. Our ongoing breeding work has, of course, a lot of its breeding and adaptation, basically garden roses. And if you look at roses, I mean, you know, people ask me all the time, well, what do you breed for? And I say, well, for adaptation and garden. And they always thinking of cut roses. I said, no, cut roses is a whole different ballgame. It's a different type of plant. They're single stems that, you know, are 60 centimeters long, and I don't want that in a garden plant because it's ugly. I mean, it's great in a vase, but not on a plant. And so anyway, so it's a completely different thing. So we have different types of roses. We have roses that have 2 sets of chromosomes and some that have 4 sets. And you really can't cross them very easily. So we have to go from one level to the other. And so that's just a lot of things we have to do. And a lot of what we're doing is combining the Basye and the Texas A&M germplasm now that has good adaptation with the Ralph Moore germplasm, which is adapted to California under dry conditions, and that's not very good here. And so a lot of it's not well adapted here. So we need to do some integration and some work to combine the really great horticultural traits that Ralph developed into the really good adaptation that we have from our work and from Dr. Basye's work. And so that's a lot of what we do these days. And we're looking for a healthy, compact plant, a lot of flowers, and hopefully no prickles, because I get all racked up when I go out that rose field, and I'm usually bleeding by the time I'm out, so I'd rather avoid that if I can. So we're working on that. A lot of resistance, a lot of work we're doing, and of course, it's all dependent on the grad students. They do just wonderful work here. We're working on black spot resistance and rose rosette resistance. So those are two major diseases. I'm also looking at Cercospora resistance, because that's becoming more of a problem. Now that we're getting better varieties that are resistant to black spot, we're seeing more Cercospora. And perhaps we just couldn't see it before because we had so much black spot. But whatever the case, so we're going to have to do some work there too. We're looking at heat tolerance, and we've had several graduate students working on this. And of course, heat affects the flower size. Here, Belinda Dream with normal heat and then heat stress. And of course, you can see the flower abortion as well that is caused by the heat. So you get less flowers overall. And then just the amount of flowers. This is in, this would be probably in August, where you have some varieties that have wonderful flowers and others that just have a few. And then there's extreme cases where the plants just don't survive. And so you get all extremes here. And here's a good example. This is in Mansfield in our plots up there. This is last August, actually. And here's White Madonna, has lots of flowers. And this is Sweet Chariot, just barely alive. But when we go back again, we'll be going back November 5th to look at things. I expect Sweet Chariot will have lots of flowers, because once it gets cool, it just does wonderful. But during the heat of the summer, it just stops. And so there's a lot of variability there. And we've had a lot of work looking at chromosomes. Again, chromosomes is to carry all the genetic material. Normally, a lot of plants have two sets of chromosomes. Of course, a lot of animals as well. And so that would be a diploid, which would be with 14 chromosomes in a rose. Now, a lot of roses have twice the number, or 28 chromosomes, which, it's just a different level. So if you cross a 14 with 28, you get a 21, which, to do crosses, they have to pair, and three just don't pair very well together. So that's a problem with the triploid. So we have to learn how to move from one end to the one level to the other, because there's some very good traits on the diploid level we want on the tetraploid level. And so we do some magical dancing to get that done. And of course, looking at plant architecture, because that's sort of what it's all about. You want a nice plant architecture. And so we have Jade looking at the various components of architecture, see if we can understand how it's inherited and how to manipulate a little better to get the perfect plant. And of course, molecular markers. I mean, both these young people are working with molecular markers. And that's sort of where breeding's going. I mean, some people think it's the same as a genetically modified organism. It's not, because you're not adding anything, you're just trying to understand what's there. And so you're basically putting tags on the chromosome. And so if this was a chromosome and you had a tag here along with trait A, which is a gene that controls trait A, you can follow that because you can follow the tag. And you don't have to look at the plant per se, you can just look at a leaf or what have you and extract the DNA. So it's just a way to get things done quicker. So as Ellen was saying, it normally takes us 3 years to evaluate something in the field for disease resistance. It'd be much better if I could do it in the greenhouse and eliminate most of my susceptibles so I don't have to take them out to the field. So again, it makes the breeding a lot more efficient, is what we're all about. And I've had a lot of students work in this area. And this is what you come out with. You do all this molecular work and you come out, this is a map of the rose chromosome, which to me is as boring as you can get. And what's exciting is if it's related to a trait. So you can see here they have a trait here and it's related to various markers and various things like that. And that's the important part for me. So I have a marker for that one trait I can follow and hopefully use it in breeding. And so again, facilitate selection and parental selection. So I can take a parent and say, well, which genes does it have? Because it's not always obvious what it has, and sometimes there's various genes controlling a certain trait. So you can look at that parent using these tools and say, well, this has gene A, B, and C, and this has C, D, and E, and I want them all together, so I cross them and try to get them there. And so that's basically what you do with this stuff. So it's molecular assisted breeding or DNA informed breeding. There's lots of different names for it. Okay, well that's sort of the quick review of what I do from a breeding point of view in the program. Now we're going to go into the Rosette project. And just as a note, if you're interested, if you like Facebook, well, we have a Facebook page for the Rose program as well as for the Rosette program as well. And so I can give you those later. You don't have to copy that.
Is that address on here?
No, it's not actually, because when that was published, I didn't have the Rosette or Rosette Facebook page. That's fairly new. I think the last 10 days. And we already have 200 likes, almost.
Wow.
I'm exaggerating, but it sounds better. Like 183 or whatever.
In other words, we need to go in and do a like.
Yes, like us, like us, and like my rose breeding program too. Because then you'll figure out what all these students are doing and when they're doing it, how they're working. Yes, the phone. Oh, that's the best.
We're going to make up a handout with that on.
Okay. Anyways, I guess you're writing it down, so I'll wait. Being polite to go.
Oh, that's okay.
Okay. So anyways, what I want to do now is give you an update of what we've been doing. We've been working on this Rosette project for 1 year. Now, granted, it took us about 6 months to get the money, but it's sort of been working 1 year, half a year without money, half a year with money, So, but they expect everything done anyways. And so, to give you some sort of background on what we're doing, and that article gives you a background on it too, so you can read about it as well. But the components of the grant, there's 6 different components: diagnostics, monitoring, best management practices, breeding, which I'm very well involved in. That's what Ellen's helping me with. Economic analysis, and of course, information pipeline, which is to try to get the information out. Of course, the Facebook page is part of that. So we're moving along. And of course, that article too. So we are trying to get things done. There's really an extensive network of collaborators here. It's just I lead it, and it's really fun because you get to talk with so many different people. But we have extension people in Texas, Oklahoma, Florida, and Tennessee working directly with the program, and indirectly we work with a lot of other people as well. Researchers in all sorts of fields, a lot of students and postdocs involved. Breeders, and this has been, to me, this was very important to work with the private breeders, because they're the people who really push those varieties out. They have a lot more resources than I can to develop varieties, and so I wanted them to be involved at the beginning. And so I work with them directly and say, well, I need these crosses done. They make those crosses. They give me ideas what crosses should be done, and then we can continue with the work, and Ellen can look at them and what have you. So we're really working very closely with these people. A lot of different rose industry, 25 different companies, of course, the American Rose Society and a lot of other societies are involved, and hundreds of volunteers. And I'll talk a little more about the volunteers later, and you're always welcome to volunteer. And of course, a lot of universities, the USDA, of course, I mentioned the American Rose Society. But again, a lot of people involved in this project. But Rosette, I don't know if you've all seen it before. Hopefully you haven't. Oh, you have, sorry. It's funny because we don't have it here, and I don't want it, mind you. But, and Dallas is not too far away. I can go up there and see it. Anyway, so it's a viral disease. It's transmitted by a mite, very small. We'll talk a little more about that. And of course, it's epidemic in the Midwest and Eastern USA. You don't see much in California, which is good, for them anyways. But little is known really about how to manage this. And it's an interesting disease from that point of view. There's 3 players in this epidemic. One, of course, is the pathogen, the virus. And we'll talk about what a virus is in a second. And the mite, it's an aerophoid mite, and it's really very small. It's not that big. It's what, a 1/10 of an inch or something like that. It's really small. You need a magnifying glass to see it on the leaf. And it floats, so the way it goes from plant to plant, if you walk, but the plants aren't touching, it spreads its legs and it just floats in the air. until it lands on something else. And so that's really how it spreads most of the time, long distance. And of course, multiflora, which was brought in here years and years ago into the U.S. It's native to Japan and China, and they use it as a conservation crop, so it has lots of fruit for the animals and hold the soil together and things like that. So they planted this all over the place, and it turned out it was well adapted. And it produces lots of seeds, so it keeps propagating itself, so it's sort of a noxious weed now. But it's there. And it's very susceptible to rosette. And mite loves it too. So it's this big body of, or reservoir of disease that's everywhere, especially in the Midwest and the East. Not so much in California. And so here you can see where the multiflora is. That's the colored area. And then there's a little in California, but we don't have the disease there, which is nice. Though originally the disease was in the mountains, in the Rocky Mountains. And actually the mite is in California, but we haven't seen too much of the disease. But it, from the early 50s to recent days, it sort of just moved down into the West Coast. So it's been slowly moving. And years ago, 3-4 decades ago, they actually were thinking of using rosette. And at that time, they didn't really know what it was. It was a pathogen of some sort as a biological control for multiflora. Well, multiflora is smarter than that, so it didn't really work, but I mean, you could infect multiflora with this. It was infected anyways. But so it's kind of interesting there. But this is what a virus is. And just, I want to give you some background to understand what a virus is, and I don't know if you do know, but what a virus does, outside its host, it can't do anything. It's got to be inside the host to function. And once it gets in the cell, and this would be a plant cell, you can see there's a little aphid here, just make that a little mite to transmit it. But once it gets in there, it goes into action, essentially takes over the cell and does its own thing. And so that's where it can reproduce. So it's integral to that plant once it's infected. It's systemic. You just can't separate it from the plant because it's there. And so that's what we're running up against, is viruses are difficult to control because once they're in the plant, they aren't on the surface. You can't put a fungicide on and kill the surface stuff. It's inside. And so it makes it a little more challenging to control. And so we're working with a lot of people. These are my virology team, and these guys are great because they know. I can hardly explain what they try to do because it's just so beyond me from the chemistry point of view. But what they're trying to do is be able to diagnose the disease before you see it, actually. Because right now, we really can't do that. And this fellow is also kind of key here. This is Dr. Tzanetakis from University of Arkansas. He's the one who first described this. And not that we didn't know the disease was there, we didn't know what was causing it. And it's only since 2011 we've known what it actually causes this disease. So, previously it was kind of hard to do anything because they didn't know what was causing it. Now we do. And so he's been very good about working with this as well. And so it was first described in 2011, and that's the picture of it. There's a little membrane and then some RNA there, which is your genetic material. Initially it was described with 4. Unfortunately, we found 3 more. So it's a little more complex than we thought. And so we're still learning a lot about this. And so, when people say, what do we know? Well, I think we know what we don't know more than anything. So we got a long ways to go. So we've figured out there's more than 4 RNAs. We have 7 RNAs. We're comparing different strains of the virus because it's anywhere from the Rocky Mountains all the way to the East Coast. It could be different. So we're looking at that. And then we're trying to figure out how to diagnose it. How can we detect it in the plant? And of course, you have to sort of tag in on something like a DNA particle, genetic material, or some of the things that they produce with respect to proteins and things like that. And so this group of people there working with me are doing that. And so currently, what we do, if we see the symptoms and see a mite, we're probably rosette. And then we can take it and we can put it through a laboratory test and we can confirm that the virus is there. But it's a very expensive procedure at the moment. And we can only detect the virus if we see the symptoms. And we know there's virus before you see symptoms. I mean, that's the way it is. And so what we need to do, and what this group of people are doing, working on diagnostics, is to develop a accurate, reliable, easy to use, quick, inexpensive test, something we can take out in the field. Something that, without a doubt, I can look at a plant and tell you if it has a virus or not. And this is essential for a lot of the work we do, even for the disease-resistant stuff, because even though it doesn't have symptoms, if it has a lot of virus, I don't want it. And we're finding out that the symptoms variety interaction is really quite amazing. There's some things that don't show many symptoms but have lots of virus. And so that's probably not good to be in your garden either, because if you have a susceptible one, well, everything else goes. And so, there's a lot of things that we need this diagnostics for. And what they're trying to do is develop a lateral flow device, which is kind of neat, because this is something you could actually take out in the field. And what it is you put your sample here, and then by capillary action, it moves over here. And this is a test line, so if in that test line it detects a virus, it changes color. And so nice and easy. Within 10 minutes, you have hopefully within 5 minutes, you have an answer. And so that's called a lateral flow device. Now, that's very similar to what they do with pregnancy tests. It's the same idea, of course, detecting different chemicals. And so you can imagine being able to go out and take something like this out in the field, and boom, you know if you have the virus. And so what they've done in the last year is they're developing new assays, and there's a lot of different approaches they're using. And they're reliable. Actually, they're more sensitive than the previous assay too. And this is they find one part out of, I don't even know how to say that number, but it's really quite accurate. So now they can detect the virus in a non-symptomatic plant, which is good, because there's a lot of nurseries that don't want to ship, the virus out, but if they can't see it, they don't know if they have it or not. And so this is going to be very useful in detecting plants that have the virus that may not be showing symptoms yet. And so we got to do a lot more work and validation of some of these tests that we're developing, as well as further adapt them to the field. And so that's what we're hoping to get to. Now, we have a big project on monitoring. This is where we're using a lot of volunteers. And again, a lot of people are involved. These are all extension plant pathologists and people that deal with or have their own diagnostic labs. And these are plant pathologists, horticulturalists as well. Again, a lot of people involved in this type of work. And what they're doing is developing, of course, training materials, because all these volunteers have to be trained on how to recognize it properly, because there's a lot of confusion on the symptoms sometimes. In extreme cases, easy. But there's a lot of things you sort of go, well, that may or may not be, because there's a lot of things that can cause similar symptoms. So again, a lot of different materials are being developed, and we're almost there to be able to release some of these. And then a reporting app. Essentially, of course, we always have to do apps, right? That's what we do these days. It's got to be able to do it in your phone, otherwise it doesn't work. But anyway, so Dr. Ong is developing a reporting app so people can record the presence and severity of the virus or the symptoms, which varieties are on, of course the GPS so you know where, so we can map it out and how it's moving or where it is, and hopefully find varieties that don't get it overall. And so that's helped me in some of the breeding work as well. So hopefully by the spring of 2006, we'll have this app, and we'll have all these volunteers energized, and we'll start getting information in. So we're getting close to that. And so this is where we have volunteers in 31 of the 48 continental states, and hopefully we'll get the rest with time. And multiple people in every state. Of course, that's what you need. You need people throughout the state. But we do know that the symptoms vary with the rose cultivar, and this is a poster that was developed by Jen Olson up in Oklahoma, and she spent a summer taking thousands of pictures of symptoms and then checking them for virus. And so these are all the various symptoms that you can find for rose rosette. So some of it you wouldn't recognize as rose rosette. And then looking at best management practices, again, a lot of different people are involved here. And what they're trying to do is understand this relationship. So what you have the virus, interacting with the rows. Of course, also interacting with the mite, which interacts with the rows. So it's not a straightforward situation. A lot of moving parts to try to understand. Try to understand the climatic effects and everything else, the genotype, the phenotype, transmission issues, the host range, and all that sort of stuff. So a lot of different things are going on in trying to understand this. And so they're looking at epidemiology and then just basically understand the mite and the virus biology, and then trying to figure out what can we do to control it in the field. Yeah, and this is both for gardeners and public gardens, as well as commercial people who have them in their selling lots, and the propagators and everything else. How do we control this thing? And so a lot of work is going on in Tennessee, and on a lot of this is Dr. Wyndham's working on this, and he's looking at planting design, effective barriers, plant spacing, how that affects the spread of the disease. So he has a lot of plots with various things. You can see these are the barriers. It's a grass that just sort of hopefully blocks that mite. It seems to be working for a little while. We don't know how long it works. So, and this is an area where there's lots of disease, and he's encouraging it too. Good plant pathologist loves disease. And they're looking at cultural control, the effect of roguing, whether you take the whole plant out or stems or what have you, how quickly you rogue. That can be very important. How can we train the landscapers? We're having some problems training landscapers because, again, there's confusing symptoms. And they don't always want to take plants out. That's the other aspect. So, again, looking at the rate of disease spread, infections in adjacent plants, and he has 5 sites. He's going to be increasing this to a couple more sites in Tennessee and Alabama. So, and these are commercial sites that they're looking at weekly to see how things go. They're looking at various miticides to see which miticides may or may not work. And there's there are suggestions on which miticides to use, but we're not sure if that really controls the disease yet. It might control the mite. We're not sure. So we got to do all this to make sure it actually works. And of course, get to the important part. I guess that's my part, right? Identify Rosette resistance, and that's turning out to be quite challenging. And this article talks about that and what we've done to sort of get started. But if you look at sources of resistance, there are several species reported as sources of resistance. And these are Asian species. These are U.S. species. And one that's adapted or indigenous to Texas would be Rosette sativa. And that seems to really have some resistance. Again, we're trying to confirm this with lots of different tests. And then there's things beyond. When you find something resistant, you have to try to figure out whether it's resistant to the virus or the mite. If the mite doesn't go on, it's going to be resistant anyways, right? But this is Rosette brachyata, which is a species from China, is resistant. It turns out to be resistant to the mite, but it's very susceptible to the virus. And so if you look at this is brachyata, you can see all the hairs there. And basically what's happening is the mite can't get close enough to the plant to feed well. It's not comfortable there, so it doesn't stay. It likes slicker things. And this is not unusual in a lot of resistance to insects and mites. But the problem with working with species, they get big. These are some species hybrids. This is a sativa hybrid. You can see that this is actually 20 feet tall. And so it's huge. It's a rose tower and named appropriately. And so to start with that level of breeding and try to incorporate into a small shrub, you got a long ways ahead of you. So really, I'd rather start with cultivated things. And so we're making a big push to understand if there's any resistance among the cultivated rows. But we have tens of thousands of cultivars. We can't look at them all. And so we said, okay, well, where do we begin? We decided that we do observational information. Jen Olson up in Oklahoma and various people in Dallas and Tennessee, they had their observations, which ones were not getting it and which ones did. And so I started collecting all that information to see if there's any consistency in it. And so we had about 700 observations up to now, and I keep adding. We have plants classified as symptomatic, suspect, or no symptoms. And now some of them we actually have some virus information on them too. And so we did all this, and we came back to this. If you look at the class of rows here, and you see the various numbers with clear symptoms, about 219. This would only be the cultivated stuff. Things that were kind of suspected. They looked like they might have disease, but we aren't sure yet. And of course, the ones with no symptoms, we still have 40 without symptoms, which is good. Of course, now we have to plant them and try to infect them and kill them, right? So they're under assault. But even with that, we got quite a few. I didn't expect quite that many. And so we have to phenotype this, which we have to decide how resistant it is. And we're finding out there seems to be levels of resistance. So we have to measure the resistances of various people involved. Of course, Ellen and Jeekins, the other graduate student working on this project. So under test, we have about 40 different species accessions. Again, trying to confirm what people have reported to make sure we have the right accession of species. Because just because they say Palustris is resistant doesn't mean that all Palustris are resistant. Could be the one they looked at was resistant, and they probably only looked at one. And so that's always an issue. But we're looking at species reported resistant. We have 200 commercial varieties in our assay or our test. And of course, we've already identified 250 that are definitely susceptible, so we don't even put them in the trial anymore. So we've gone through at least 500 different things. We had another 50 or so to go. Probably end up to about 100 or more this year to put out there. But again, you get a wide range of diversity, all types of classes and types of roses. And I've talked to breeders and said, well, give me the most diverse stuff because I want to see if anything has any resistance. And of course, if they are, they're going to be used in breeding quite a bit, I suspect. So here's a shot of just some planting the germplasm out there in Tennessee, trying to get that resistance. And again, this is an isolated plot. They're augmenting as much as they can with Rose Rosette. They introduce susceptible ones that are infected, try to get as much disease as possible. And again, just another shot of that. And so they're doing a lot of field screening. And what they do, they plant it, and then they get infected things and take shoots of that, put them on the uninfected, try to get it all infected with mites and disease. Again, we want to know if the plant can resist it, either by resisting the mite, or if the mite inserts it, it's resistant to the virus. And so various things. So once we see something that's resistant or tolerant, we're going to have to do grafting. And with grafting, if you graft a rose with an infected bud, the virus has direct path into that plant. So that's the way to test whether it's resistant to the virus. And hopefully it is, because that's what I would prefer rather than just resistant to the mite. So, but we have to distinguish between these different possibilities. We're also doing a lot of other phenotyping. Of course, we're phenotyping for flower types and Cercospora and black spot resistance and all this material. And this is our trials in Texas. Overton got planted this spring. We had problems with irrigation, so ours are still in pots. Hopefully this fall we'll get them planted and get those out. Again, we have 250, 300 different varieties we're trying to put out there. And then the genotyping. We talked about molecular markers. There's lots of different molecular markers out there. And this is the old-time molecular marker. Those are microsatellites. And again, working with Jeekin and Ellen and Mandy. Of course, Dr. Klein works with us. She's the molecular side of it. I'm just a plant breeder. And what we're trying to do is there's a new technology out here to really develop a lot of markers. Previously, when I worked and made maps, that one map I showed you took us 2 years to make, and a lot of work. Hopefully with this type of approach, called genotyping by sequencing, or next generation sequencing using that technology, we can develop more markers, more tags on those chromosomes a lot quicker. And hopefully in 2 to 3 months, we can develop a map that's even better than what we could do in 2 years previously. And so that's what we're hoping to get. And this is what we've done. This is preliminary work. This is the old map where we had on this chromosome 17 markers. Now, on the new map, we have 125. The 17 took 2 years. This took 2 months, once we figured out a few details. And so we can go a lot quicker and get more markers, so we have a better chance of finding a marker that's associated with the trait that we're interested in. and then use that tag, let's say this cool 2295 1232, so we can follow that gene. But they don't have really good names yet. We got to work on that. But anyways, so that's sort of the idea. And of course, I'm working with a lot of breeders, and this is the range of breeders I work with. Actually, there's one more breeder who joined up, worked for Ball. Again, creating the rose crosses we need to do the analysis. And so this past year, we just got started. So we did about 3,000 pollinations, which, it's a good number. We did 1,800 with Palustris and Blanda, which are species that are thought to be resistant to the disease. Unfortunately, the results were not good there. We got very poor set and poor seedling recovery, so it might not be the way to go. That's what you learn. That's why working with species is sometimes very difficult, because they're not always compatible with the ones you want to cross with. Now, we did a lot of other crosses with Satidra, and they seem to be taking a lot better. Carolina derivatives, and among cultivated ones that we think are resistant. So we're just in the beginning stages here, and we should have, a couple thousand seedlings this coming year to look at. And then what we want to do is take all the phenotyping, essentially, is it resistant or not, put that with all the markers, and then there's some software we can put all that information together, and it tells us which markers are associated with traits. And so that's sort of what we want to do, looking at the pedigrees and all this information. So a lot of different things to do. We work with several people in Netherlands on the software and the analysis aspect of this as well. And then the final, or one of the other parts, not the final one, is the market and economic analysis. And we have three fellows, economists here working with us. Again, we want to understand how important this is from an economic point of view. Because we all say, it's horrible, it's killed my plants. But when you talk to politicians, they say, well, how much is it worth? Well, it's a good question. And so, usually scientists say, we don't care, it's just horrible. But, so these guys are going to tell us how much it's worth. We hope anyway. And they're doing some interesting things. You see, he's hooked up with electrodes, and they're trying to measure his brain waves to see if he's sane, I guess. I'm not sure what. But they're going to look at consumer preference and a bunch of different things, again, to quantify the relative importance of various traits, disease resistance being one, but they're going to go beyond that to sort of understand the rose as an economic crop. Looking at barriers in the marketplace to get these roses out. Once we develop resistant ones, what do we have to overcome? What do we need to really make them worthwhile? Because we can get something resistant, but if it doesn't have everything else, no one's going to plant it. I can tell you don't want to plant Rosa Satidra. It's resistant, but it flowers once a year and it just takes over your whole yard. You don't really want that, I suspect. And then look at the socioeconomic impacts of Rose Rosette. I'm not quite sure how they do this, but they tell me they can. I believe them. And so they're using a lot of the biometrics here. They're looking at brain waves and galvanic electroconductance to see how emotional you are. They tell me all this works. Read your face, so be careful when they test you. They're going to read your face too. And all sort of visual attention behavior, all sorts of behavioral things to understand what's going on in your mind. And how do you study you more, basically. And of course, we want to study more roses. So this is good. And then we have the information pipeline. These are extension people that are working with us in developing materials to get things, the information out. And of course, they're helping with the Facebook pages. There's a Twitter account, though I don't know how to use Twitter yet, so I don't know the account. Writing publications, extension bulletins, all that sort of stuff. And of course, getting students out to talk about it and tell it at the American Society of Horticultural Sciences meetings. As, of course, we had several other students go to, always talking about various things that we do. We're getting press releases out. We're developing online resources for training and information and various things. So we're trying to get all this out. After a year, we're getting close to start pushing information out. And of course, again, the grant is complex. There's a lot of different things we're trying to do. And this first year has been sort of, trying to push everyone into it, and let's just get things established. And The diagnostics people have gotten a lot done because they do everything in the lab. It's a lot easier. If you get in field stuff, we're just trying to get things planted. It takes a while to get going. But we're getting there, and next year is going to be really exciting. How about current management recommendations? I'm sure you're all interested, right? There's two ways to do it. You either exclude the virus or reduce the mite spread and mite populations. That's the two approaches you have. Okay, so obviously only plant roses that don't have rose rosette. Simple. Well, if you could tell. If it's symptoms, you can tell. If it's no symptoms, well, you really can't. And so one of the things we're doing is trying to figure out whether the infected plants are being shipped around and see if that's a major issue or not. So that's one of the things, because we haven't had a way to tell yet. Eliminate rose rosette infected roses from 100 meters of your garden. Well, if your neighbor has them, well, you're going to have to talk to them before you rip them out. But it's good suggestion, because that you don't want anything near your nice roses. I mean, there's no doubts there. Monitor weekly and eliminate row out plants with symptoms. So I missed a word there. But they're talking about monitoring weekly, and that's a hard thing to do. And in South Carolina, they're doing this. No, not South. In Tennessee, they have a public garden, where they monitor weekly, and every week, if something's showing any symptoms, they think it might be rose rosette, they take it out. And by doing this, and this is a very high pressure area, there's a lot of rose rosette in the area, they have to replace maybe, he said, 3 to 4% of the roses every year. But he said, that's not a problem. I mean, you're talking about 1,000 roses in this garden. So taking out 30 or 40, not a big deal. And that way it doesn't get started. And so you have a real nice rose garden anyways. And he says, he does this, and the curator probably doesn't even notice he's taking things out. I mean, so, it works to do it that, but they're monitoring weekly. And so that's key to get them out quick. If you want to reduce the mite spread, you don't plant them real close together. And a lot of these commercial plantings are so close together, I mean, the mites just walk from one to another. They don't even have to fly. They just walk. If you remove it, you need to bag it. And the problem is, if you don't bag it, those mites fly. and they stay in your garden. So you have to take it and actually put it in a plastic bag and get it out before those mites spread. Because once that plant starts desiccating, those mites go up and look for a wind or a breeze to take off. Do not use blowers. And you'd be amazed how many people use blowers to clean out their roses or any landscape situation. If you use a blower, well, you're blowing the mites all over the place too. And so that can be an issue. Do not go from infested garden to another garden. And you say, well, why not? It's virus. It's not going to attack me. Well, the mites are going to be on. That's key. That's not the virus per se. That's not going to be carried on your hands or anything, but the mites are going to be in your clothes or what have you, and you go to another garden and a gift that they don't want. Prune heavily in the winter. You do this, you eliminate mites, because they overwinter on the canes. So if you eliminate it in the winter time, so you don't have any mites or less mites to start with, you're in better shape. And then dormant or summer oil may help. It seems like oil does decrease your mite populations. And there's certain oils you can apply that are safe and work fairly well. And so that's sort of the current situation. Yes?
How long do they live not on a rose, but say on your clothes or on a shovel or something?
They will, as far as we know, and this is still a little guesswork, 1 to 2 days is what they're going to survive. So it's in terms of a couple days rather than hours. And of course, or months, yeah. No, it's not months either. But, if they're on your clothes and they want to fly, well, it's easy for them to fly too, So. And, the worst thing I can think of is a landscape person pruning out these roses, putting in the back of their pickup truck and taking off.
Without bagging.
Without bagging. I'm sure that happened, but nobody knew. So it's not their fault yet. They don't know. They're beginning to learn, and I guarantee you there's a lot of interest in the landscape industry to be able to manage this very, very quickly. Yeah.
If the plant becomes infected and you pull it out, is the soil under it?
No, the virus won't be in the soil. though you have to be careful. And some people say, oh, you should take all the roots out. Well, probably not, because again, the virus may be in the roots, but it's going to die with the roots. So it's not going to go from a dead root or a dying root into a plant. But if you leave part of the stem there and it shoots up again, you're going to have it. So you have to make sure you get all the stem tissue out, so there's no more shoots coming up. But you don't necessarily have to take all the roots out. Okay? So that's important too.
Where you said prune heavily in the winter, do you mean around Valentine's Day or winter?
Yeah. I mean, you can do it before things start growing again, because the mites won't be very active when it's cold, because they're cold-blooded, so they just hunker down. Okay, well, again, we have lots of collaborators. And of course, these are some of my students. And this is really the reason that makes it so much fun to work with students. They're really, and I do appreciate your support of students. It's been wonderful to have this scholarship available. But you can see they're all still smiling. This was in August up in Mansfield, after being out there all day looking at roses. And it was, 95 to 100, somewhere in that range. And so, to be able to smile after that, that's amazing. And so I think they were happy because they're going home too, but whatever. And here's a group that went to the national meetings as well. Of course, Ellen's there, and then 4 other graduate students went. So we had good representation at the national meetings as well.